Hello. It's 4 o'clock now, so we might as well go get ourselves started. So it's my distinct pleasure today to introduce our speaker, Dr. Dave Marcinek. Um, Dave is, I've known Dave for probably 10 years now or so. And he's a professor in the Department of Radiology, and where he studies mitochondria, where what he's going to be talking about the interplay of mitochondria and aging, which is something that I also think is very cool. So Dave got his start actually, his PhD in physiology at Stanford, and then he actually moved here to do his postdoc back in 2000, and has remained here ever since, kind of like me. And he's built this program looking at mitochondrial activity, and he's going to be talking about that and some nice, really cool work and interventions in um, in mitochondrial dysfunction and aging that can kind of help ameliorate that. So, without further ado, I will turn it over to Dave.
All right. Thank you. Thank you, Scott, and thanks for the invitation to speak to this group, and thanks for coming out in the afternoon here. So yeah, Scott said, so I've been here for 24 years, right? Mostly studying mitochondrial function and energetics, and we came in with a real focus on the energetic side of things, and that's where a lot of my work has been focused, and that's what I'm going to mostly talk about today, and then at the end talk about how we've kind of we're trying to mature our view of the mitochondria. So thanks for coming out, and I do want to say right up front, there's a bit of a bait and switch. There is going to be some bedside. There's going to be some translational work in here, but there's not as much clinical translational work as I thought at the time, because I wanted to add some cool stuff that we're doing at the end. So I pulled some of that out. So, here we go. Okay, got to hit it a little bit harder. Okay, so this is my disclosure slide. Do I have a cursor? Yeah, here we go. So the work I'm going to talk about today is mostly focused on our work we've been doing with elomeprotide over the last 10 or so years, but we've had a lot of other collaborations with industry partners and various foundations. So for the outline, what I'm going to try to share with you today, first I'm going to spend a few minutes just talking kind of big picture mitochondria, muscle, and how it relates to healthy aging, and then really kind of get into some of our work on using targeted interventions to improve mitochondrial function and improve health in mice, and then move to some clinical trial support where we actually translated that mouse work to actually a clinical trial in older people, and then kind of move back, taking one of the things we learned from this clinical trial, taking it back into the mice to try to get a little bit more understanding of the actual mechanisms of things that are happening. And then right at the end, I should do this so the virtual people in the virtual world can see. At number 5 here, this is a new approach we're doing with a collaborator in genome sciences to kind of taking a step back and kind of looking at a big picture of mitochondria and aging. So anybody that has been to more than 2 aging talks has seen this slide. And I just announced in my lab yesterday, this is the first time that I have ever shown this slide in a talk. Almost out of stubbornness. But basically, this is a really valuable paper, kind of laying out the hallmarks of aging. These are some of the basic principles that contribute to the biology of aging. And it won't be a surprise to anybody that this is where we focus most of our energy. One, because the mitochondria are just really cool, right? They're this really cool self-contained organelle. We like to think of, they control most things going on in the cell. But from a more practical perspective, really, they sit either at the center or connected to all of these other hallmarks. So by better understanding what's happening in the mitochondria with age, and then actually intervening to target the mitochondria, we feel like we can affect a lot of these other hallmarks. So another part of what our lab does is study skeletal muscle aging, or sarcopenia. So that's the loss of muscle mass and function with age. So when you think of aging or an older person, this decline in muscle tone, muscle function, muscle mass is kind of one of the first things we think about. It's one of the most identifiable aging phenotypes. And this loss of the ability to generate force and power is a real contributor to this increased risk of falls, as well as this exercise intolerance and frailty, which then is associated with comorbidities and just generally poor quality of life. And lastly, for this slide, one of the main points, we see a lot of fat infiltration and a lot of metabolic defects that are associated with skeletal muscle aging. And this is true regardless of activity level. So this person on the left, Ed Whitlock, was a marathoner. He was running marathons into his late 80s, right? Super healthy guy. He's clearly got aged muscle, right? So although physical activity is critical to staying healthy, muscle ages regardless. And you can see this in several studies now have published data similar to these, where they look across, let's see if I can coordinate this here. They look across section of ages here, and then they look at thigh area. Yeah, I'm not going to do this. You see this decline with age going, in both muscle area on the right, on the left, and muscle strength on the right. And these slides, they showed a gradual decline in both. I prefer to think of this as there's a plateau up until about the mid-50s before you really start to decline. And as I'm, in my mid-50s, I'm feeling more and more strongly about that. But regardless, this clearly shows that there's a clear decline in both muscle mass and function, and the function kind of decline is a little faster than the area. And this is a multifactorial condition. A lot of things feed into this decline in muscle function with age. But where our interest in mitochondria and muscle converge is that we really think that the mitochondria kind of play a role in all of these things. Is there any ones that mitochondria sort of all of these things cause sarcopenia, and the mitochondria play a role in all of these things. And so the way we like to think of mitochondria in the lab, it's either a cause or a consequence of most things that happen with age, right? So it may not be the primary driver, but it's an amplifier of the pathology, right? Yeah, I wouldn't say that I wouldn't say there's one cause. There are nodes where you can intervene, and I think I'll talk about what one of those nodes might be today. Yes. But this interchange between mitochondria and muscle and aging is borne out by several large studies. And this is from the SOMA study relating the mitochondrial energetics in skeletal muscle with cardiorespiratory fitness and mobility. This other one is relating it to walking speed. And there's more and more coming out from these large cohort studies where they're actually looking at the relationship between skeletal muscle energetics and other factors of health. So in this case, they're looking at cognitive impairment and biomarkers of Alzheimer's disease and relating that to muscle function. Again, this is a large cohort study where they're looking at relationships, right? So there's not a cause and effect arrow here, but there's a clear relationship. And then this is a study from, I think, 20 years ago where they looked at people that had really high metabolic fitness, which is these dark gray bars, and then low metabolic fitness, these light gray bars, and looked at the risk of death across these 6 chronic conditions, and see that going from high fitness to low fitness almost doubles your the risk of death across the span. So with all this in mind, what we're trying to do is really ask the question, or can we target the mitochondria to improve muscle function and general health? So the idea is we're trying to see, can we come up with interventions to go from here to here, getting out of this disability threshold? So the questions, I think I've kind of beaten this to death. Are the mitochondria a promising target for improving muscle function? And then can we use mitochondrially targeted interventions to learn more about the basic biology of aging? So it's not just a one-way arrow, but can kind of circle back from the clinical trial back into the mice. So in order to pursue these questions, we were fortunate enough about maybe 10 years or so ago to start working with a compound called elamipretide. A lot of you guys have probably heard about it. Several people here work on it. Mariah talked about it about a month and a half ago in this very room. But it's this tetrapeptide that was discovered accidentally that associates with the mitochondria by interacting with this fatty acid called cardiolipin on the inner mitochondrial membrane. This is a really important component of the mitochondria because it helps to structure the cristae membrane or the inner mitochondrial membrane while the business part of the mitochondria works. And these cardiolipin molecules also interact with all of the complexes of the electron transport system and help to sort of arrange these protein assembly into supercomplexes to make more efficient mitochondrial function, as well as altering the properties of the membrane. And this is some work that's really just come out in the last couple of years, which is really neat. So now kind of jumping into our work. So we use a lot of the energetics data that I'm going to talk about today is using an approach we call in vivo metabolic spectroscopy. So we want to be able to study the energetics in the context of the living functioning animal. So this is an anesthetized mouse in our magnetic resonance coil. So we use magnetic resonance spectroscopy to study ATP production in the intact skeletal muscle. And then we can use optical spectroscopy to measure oxygen consumption. And from these data, we can measure the maximum ability of the mitochondria to make ATP, as well as the coupling ratio, or the ATP produced per oxygen consumed, or the P/O ratio, which is a measure of the efficiency. And you can think of it as a measure of proton leak of the mitochondria. So this system was designed by Mike Siegel, who's a bioengineering graduate student at the time. There we go. And so our first experiment we ever did with elamipretide, came out of a big collaborative meeting, and it was almost like, okay, well, if this is how it works, then this should happen, right? So Mike took young and old animals, gave them an injection of elamipretide, and then measured their in vivo mitochondrial function an hour later. And so what he found was when he looked at the young and the old, there was a significant decline in the mitochondrial efficiency. So the P/O went down, indicating there was greater proton leak. The mitochondria were leakier. And after an hour of treatment with elamipretide, this went back up to young level. Same thing when we looked at maximum ATP production. This went down significantly with age, back up after an hour of treatment. So these are really rapid changes that he was observing. And then later, Matt Campbell, who's now an instructor in our lab, did a similar study to sort of build on this, and then he did an 8-week treatment and basically saw the same thing. Reduced P/O ratio with age, restored after 8 weeks of elamipretide treatment. Same thing with ATP max. Then Matt went on to look at skeletal muscle function. So here we're looking at the tibialis anterior. And in this case, the muscle is exposed, we tie off the tendon, and then we can stimulate the muscle directly. So this keeps the circulatory and the nervous input intact. And then he looked at animals, older animals that were either treated with a control or elamipretide in blue, and saw that the elamipretide treated animals were more fatigue resistant. So associated with the improved energetics, we saw that the muscle had greater endurance capacity. And this translated to treadmill running time. So these are measures at baseline in the old animals and the young animals. And then after 8 weeks of treatment, there was no effect in the young animals, but in the old animals, they ran significantly longer. And in spite of these differences up here, we didn't see a change in the maximum force that the muscle could produce. But more recently, Gavin Farrow, who's a postdoc in the lab, did a study where he looked at the gastrocnemius muscle, stimulated the muscles in vivo, and measured force over this force frequency range. So this shows sort of early, low levels of muscle function. This is where a lot of your sort of activities of daily living happen in here. Very seldom do we function at maximum force production. So what he saw in the old animals, there was a significant decline in this maximum force. But in the old animals treated with elimipritide, where you really saw the difference was in the submax force production here. So in spite of not seeing a change in the max, in sort of this functional range, there was still an improvement in muscle force. And these next two slides just show that these effects sort of are broader than muscle. They also expand to the cardiac and kidney, which you saw about a month ago. So these are some data by Ann Chow when she was when she was a postdoc in Peter Rabinovich's lab, where she looked at aging mouse cardiac function using echocardiography, and saw that the old animals had significant diastolic dysfunction here, shown by this low E/A ratio. And then after 8 weeks of treatment, this was significantly improved towards the young level here, shown in orange. And then similar for the myocardial performance index, which is kind of just think of this as an integrated measure, incorporating a couple different parameters of function. And here, 8 weeks of treatment brought this back to a lower value, which is more towards the young. In the last year, Gavin Farrow also sort of expanded our ability to do echocardiography using this global longitudinal strain analysis, and looked at systolic function. So this systolic strain is a measure of ventricle deformation during the systole. And this goes down with age, and after treatment is restored by SS31. There we go. Yeah. And this next slide is you've seen before, most likely. But essentially, the take home is that Mariah showed back when she was a postdoc, I believe, that animals treated for 8 weeks also had less kidney injury. So this was reducing the kidney pathology associated with aging. So this is a measure of glom injury. 8 weeks later, this goes up. It doesn't change in the animals treated with elimipritide. And these are all things that our lab is currently focusing on right now. So I kind of went through this first part relatively quickly. It's all published data, and probably a lot of you have seen some of the data before. But basically, to summarize, that these age-related mitochondrial energy deficits can be reversed with elimipritide treatment. And this is associated with improved function kind of across multiple physiological systems. And what was really striking to us was that these rapid improvements in energetics are similar to what we saw after 8 weeks, which really kind of shakes up how we think about an intervention might work. Most things that target the mitochondria target something upstream and either induce mitophagy or induce biogenesis and require protein synthesis and activation of pathways, where whatever's happening here seems to be happening relatively quickly. So taking this idea that we're seeing these rapid changes after quite a bit of lobbying from me, I worked on STELL for a while to get them to try to support a study where we capitalize on this acute effect. So finally, they supported a study, a phase 2 randomized controlled clinical trial of elimipritide in older adults. So what we did, and the study was led by Kevin Conley. So we screened people for average to low mitochondrial function using the criteria of an ATP max. So this is maximum ATP production below 0.7 millimole per second. You can see this is the population from several clinical trials. You can see that this 0.7 kind of differentiates right down the middle from the high functioning and the low functioning. Then we gave these people a single dose of elimipritide and measured in vivo energetics again and muscle fatigue resistance. Immediately after the infusion of the drug, and then 1 week later. Oh, and this is just the flow diagram. So again, we used a phosphorus NMR to measure the energetics, and we were looking at the hand muscle here, which is this muscle that causes your finger to move like this. And see it there we go. And I'm happy to answer questions about why we studied that muscle if you want after. But then we looked at mitochondrial energetics right after infusion, and this is a plot of the change in the ATP production post-infusion compared to baseline. And we saw an increase within a couple hours of the infusion. And then after 7 days, this went back down to baseline level. So again, it's getting in there, doing something fast, rapidly doing it, and then at least in terms of the mitochondrial ATP production, then it's washing out. But we also looked at fatigue resistance. And this is our homemade instrument for measuring muscle force and fatigue. And so the assay here was just asking subjects to repeatedly do voluntary contractions at 70% NVC as long as they could keep it up. And then once they couldn't do it anymore, that was the end of study shown here. And our measure of fatigue in this case was we just counted the number of contractions they could maintain. And so interesting, what we found was there was no effect immediately after infusion, but when we looked a week later, muscle contraction continued to improve and fatigue resistance continued to improve. So to summarize this clinical side of things, basically the take home is that these rapid improvements in energetics that we saw in the mice translated to older individuals. So the next question is a question that, we were wondering, but every time I gave this talk for quite a while, people asked the question, so we know that elimiprid interacts with this membrane, but does it actually sort of do sort of a more typical drug targeting where it actually interacts with specific proteins, right? So in order to address this question, we started a collaboration with Jim Bruce. Jim Bruce and Juan in Jim's lab in the UW Genome Sciences. And they've developed a really cool approach. It's called cross-linking mass spectroscopy, where they can actually look at how proteins interact with each other, and in this case, with small molecules. So in this case, we used the biotin labeled SS31. And SS31 is another name for elimiprid. And then took advantage of the presence of this lysine residue in the normal structure. So we took the hearts from old mice, isolated the mitochondria, incubated them with this biotinylated SS31, then incubated them in the presence of these, oh my gosh, I'm going to forget the name, protein interaction reporter molecules that Jim's lab has come up with. And these molecules form cross-links with lysine residues. So we capitalize on the fact that there's a lysine in SS31 and there are lysines in proteins. And so with this reaction, any molecule of SS31 in close proximity to a lysine in a protein is going to get cross-linked with this reporter. Then you do a sort of a typical protein digestion, and then do an enrichment by pulling down with the biotin tag. And then what you're left is this complex with your biotinylated SS31 and your peptide. Then you do your mass spec and your normal analysis, and then your structural analysis, and identify what proteins interact with this drug. So what we found was there were 12 specific mitochondrial proteins that bind with the drug. And these are associated with oxidative phosphorylation and ATP production, or alpha-ketoglutarate processes. And what was interesting, all of these proteins have been reported in literature to associate with cardiolipid. And this is kind of where they fall in the cristae membrane. So these are the complex 2 sites, complex 4, the cytochrome oxidase, the ATP synthase, a couple matrix proteins. But we, for multiple reasons, were particularly interested in this ADT1, which it's also known as the adenine nucleotide transporter. So this protein is responsible for bringing ADP into the matrix of the mitochondria and then pumping ATP out to the cytosol where it can be used. And what's really cool about this approach and this mass spec that they do is that not only can you identify the peptides that interact with the proteins that interact with the peptide, but you can actually identify the sites. And so you know where the peptide is in the protein. So what they found is SS31 interacted with lysine 272 and lysine 33, this peptide. And then Juan did some modeling to identify the most likely position of the peptide in this protein, and basically found that it sits in this channel. So as I mentioned, the adenine nucleotide transporter is a transporter, brings ADP in, takes ATP out. But also one of its functions or side effects is it's also a main proton leak channel in the mitochondria, because this channel can open up and be permeable to protons, especially in the presence of fatty acids. And so based on the modeling, we were hypothesizing that SS31 can come in here and alter the structure of this transport channel to inhibit the proton leak. Then also, well, the next question I guess we want to ask is, does it also affect the ability to transport ADP? And the modeling studies and the structural studies are ongoing about that. But we wanted to look at it, go back to the energetics, right? Back to where I'm more comfortable. So we went back and looked at some of our energetics data. So these are reanalysis of our in vivo spectroscopy. And rather than looking at the maximum ATP production, we wanted to look at the sensitivity to ADP. So you can think of this as the ability of the mitochondria to respond to energy demand in the cell. And so reanalyzing these data, we saw that, so in the gray is the young, the black is the old. So we see a shift of this curve indicating that the mitochondria are becoming less sensitive. But then in the old treated with elamipretide, we see this opposite shift indicating a significant increase in the sensitivity. And that's just quantified over here. Now, the more rigorous scientists out there are going to say, hey, wait a second, you're drawing a line through a couple points here. I don't really believe that very much. So recently then, again, Gavin Farrow, there we went back and looked at this in permeabilized muscle fibers and isolated mitochondria. So in this case, he isolated muscle from these 26 and 28-month-old wild-type mice, either treated the tissue during the permeabilization or during the mitochondrial isolation with elamipretide, or treated the animals for 8 weeks and then did the permeabilization or the isolation. And again, found that compared to the wild type, the elamipretide treatment significantly increased the sensitivity of these mitochondria to ADP. So that is, as ADP increases this way, their rate of respiration increased more quickly in the presence of elamipretide. So this would be consistent with better transport of ADP from the cytosol into the matrix where it can be used. But because this respiration is a complex process that involves the electron transport system, ATP synthase, and the ANT, Gavin wanted to know specifically whether this was the elamipretide was affecting the ability of ANT to actually transport ADP. So here he teamed up with Ian Sweet to actually measure the import of radio-labeled ADP into the matrix of the mitochondria here, and then could also do this in the presence of a specific ANT inhibitor. And so what he found when he looked at both in skeletal muscle and heart mitochondria from old animals, that in this range of this low ADP range, this is a sort of moderate energy demand of skeletal muscle range of ADP, there was a significant improvement in the ability of the ANT to pump ADP into the matrix of the mitochondria, suggesting that this is a main driving force behind this increased sensitivity of the mitochondria to respond to energy demands. And these data suggesting that ANT, or showing that ANT actually is transporting ADP better, are also consistent with work done by Huilang Zhang when he was a postdoc with Peter Rabinovich, where he looked at cardiomyocytes isolated from old and young animals here, and then incubated the old cardiomyocytes in the presence of elamipretide, and measured their respiration in the seahorse. And what he found was that they were going from, there you go, going from the young to the old, there was a significant increase in proton leak, similar to what we saw in vivo back in the beginning of the talk in vivo. But then when they were treated with this peptide for, I think these were an hour or two treatment, really, really short-term treatment, this reduced this proton leak. And also when he used ANT-specific inhibitors, this also prevented the proton leak, indicating that this is happening through the ANT. And interestingly, they did not see any difference in maximum respiration. Again, supporting a role that, maybe a lot of the energetics improvements that we're seeing with this treatment are due to changing the sensitivity of the mitochondria to energy demand. And so to conclude here, basically, going back to take a closer look at our energetic stuff, and then this cross-linking mass spec really suggests that elimipretide is interacting with specific mitochondrial proteins, and that this direct binding of elimipretide to the ANT may be an important part of its mechanism of action by increasing sensitivity to ADP and reducing proton. So now in the sort of the last, couple minutes, I'm going to switch gears a little bit to talk about a slightly different direction the lab's going in, but a related direction. So I spent a little bit of time in the beginning talking about how mitochondria are involved in everything. They're so important, but then I spent the last half an hour just talking about the energetics, right? Really focused on the energetics. And that's kind of this powerhouse view of the mitochondria, right? They make ATP, and as a byproduct, they make oxidative stress that can do bad things. But in fact, I think now we really recognize that mitochondria are really a nexus for cell physiology. They're involved in a lot of things happening. Integrating signals from the environment, and these are just a few the things that the mitochondria are controlling. So it's really important to kind of take a broader look. And I think one of the limitations that some of our mitochondrial research has been under, and I think a lot of mitochondrial research kind of in the field, whether you're an energetics person, whether you're an imaging person, a mitophagy person, a ROS person, if you have a hammer, everything looks like a nail, right? And that our hammer was looking at the energetics. We were really good at measuring respiration, really good at measuring ATP production, and with a little help from our friends, pretty good at measuring redox stress, right? But to kind of beat metaphors to death here, this is kind of a limitation to this single approach, right? So you've got all these people that have their specialties, the blind scientists describing different aspects of the mitochondria, thinking it's, and it's funny because you go to meetings and people argue about the most important part of the mitochondria, and whether we should be thinking about this or this, or, and it's basically what people can measure, right? And that's what they want to do. So what we wanted to do is we wanted to take a step back and try to come up with an unbiased tool to kind of look at the state of the mitochondria, in this case aging, but under different disease conditions. And this was from a review published, I guess, last year. It's becoming pretty highly cited. But this just breaks down the kind of different levels at which we can look at the mitochondria, starting from the molecules up to sort of protein expression, activities, structure, and then behaviors, and then ultimately how it affects health status, right? So as you can see here, this is moving from furthest away from organismal function to getting closer to organismal function. So what we wanted to do, we wanted to think about how could we sort of address these this multi-scale, multi-level problem, and we decided we want to function kind of in this area, right? So we can look at the protein expression, and it's clearly a critical component. We need to know about enzyme activities and transport. We need to know how the proteins interact with each other, how they can work together to form structures. So ideally, whatever we did would be able to assess protein expression, protein structure, as I said, the interaction or assembly, especially in the case of the mitochondria, where they form these super complexes that are really drive efficient function, and then as well as regulation by post-translational modifications, and then also learn some information about protein and ligand interactions. Well, fortunately, we were still collaborating with the Bruce lab. So it turns out that this mitochondrial protein interactome can address all of these things by looking at how the different crosslinks, these different proteins change. So you did a study with Jim and Anna in Jim's lab. And again, this on our side of things, Gavin was the main driver for this, where we, and the process here, sorry, we isolate mitochondria and then we can split them. We split them and we look at function, right? So we can get an idea of, one, whether they're functional after the isolation, but then how are they changing sort of at the energetic molecular level, but then take this through this similar cross-linking mass spectroscopy approach. And in this case, though, the novelty or the advance that Jim's lab had come up with is they used two different of these types of these PIR molecules, and that can be distinguished in the mass spec. So we used one of them to cross-link the young mitochondria and one of them to cross-link the old mitochondria, and pair them up, run them through the mass spec, and then what we get is the ratio of the old to young on the intensity of any given crosslink or any different protein-protein interaction. And so from that, we get information on conformational changes and different quantitative networks, basically all the things that I mentioned in the previous slide. So last year, we published a sort of a proof of concept paper on this in Nature Aging, where we had 4 pairs of animals, and we just showed sort of some broad differences and aligned those with some of the changes in respiration. Now so these are some data now in process, where again, Gavin's leading this, working with Nate, I think I'm blocking Nate, in the Bruce lab. In this case, we're using 6 old-young pairs of males and 6 old-young pairs of females. Use the same cross-linking mass spec approach that I discussed on the previous slide. And so this is the volcano plot showing the log2 ratios of the old-young. And all of these are specific single interactions of peptides. They could be intra-peptides interactions, or they could be inter-peptide interactions. So the intra-peptide interactions are going to giving information on protein structure, and the inter-peptide interactions are giving information on protein assembly. And so these are the changes in the female 6 pairs and the male 6 pairs. So this is very much a work in progress. These data are currently being analyzed. But in the interim, what Gavin did was he took all of these changes and calculated an average log2 change for the intensity of crossing. So basically, we're saying the higher the average change in this crosslink with old over young, the greater the global change in how the structure of the mitochondrial protein. And then he looked at this in relation to gastrocnemia's mass and force. And in the mass, I was actually surprised that he saw such a good correlation with so few animals, and still related to muscle force, but a little bit less so, suggesting to us that we're looking at some biology here. Right? There's something related to muscle function. Yes. Yeah. Oh, yes, okay, yeah. Okay, well, I'll do my best. Basically, so the question is, so it's clear down here in this plot that the muscle mass and force are separating the sexes, but are the degree of cross-linking also separating the sexes? And the answer is yes, and we'll see that much more on the next slide, right? Yes, okay, so this was looking at how this relates, this general measure of, almost esoteric measure of mitochondrial changes relates to muscle function. So we also wanted to take another step back and kind of look at, does this relate to the health status of the animals? So to do this, Gavin has been putting a lot of effort and doing a lot of analyses on using this frailty index, which is this multi-parameter assessment across multiple systems. It's totally not invasive. It doesn't really disturb the animals much. So it's really great to do to get just a general health status of these animals you can do at longitudinal. And these are a list of all the different parameters. I'm not expecting anybody to really take anything home other than that some of them are age dependent and some of them are not. So they have varying degrees of information. But when they're summed together, they go up with age, as you expect. And the females have a higher frailty index than the males. So the female health status at a given age tends to be lower than the males at a given age. So then we wanted to take a look and see how does the skeletal muscle mitochondrial interactome relate to this health status measure for these mice. So in answer to Mariah's question, so this is the average value of the change in the mitochondrial interactome in the females and the males. So the females had a significantly greater alteration of mitochondria with age than the males did. And they also had a significantly greater frailty index, which is indicative of poor health status, more frail. And then when looking at the individual animals, again, you haven't found a relationship between the degree of restructuring of the mitochondrial proteome, change in the mitochondrial proteome, and health status of the animals. So I was really excited by this. I think this is early in the process. But so one of the things that Gavin is doing is trying to improve on this frailty index and as a predictor of biological age, right? Because as I mentioned, I think only 23 of the 31 parameters actually change with age, and they change differently in the males and females sometimes, right? So what he's in the process of doing now is using this huge training set that he's acquired over the last couple of years. So he has 244 training mice, males and females, and he's using a deep neural network model to predict a predicted age and relate that to the chronological age of the animal. And what he found was pretty reasonable correlation. So kind of validating that we're measuring something that relates to chronological age. And this is similar to kind of what you see with epigenetic clocks too, this level of variation. So then the next step for this was to look at this predicted age related to this change in the mitochondrial protein interactome. Again, we see this really what I think is a pretty neat correlation, suggesting to us that maybe we're onto something here, kind of looking at this, taking this global view of mitochondrial protein, the structure of the mitochondrial protein, and relating that to this overall health status of the animal. Oh, and I want to make the point here that all of these animals on here are the same age. So this range in predicted age represents changes in what we are calling sort of biological age. So some are aging faster, some are aging slower. We could do that, not for these animals, because these animals are involved in a study. Then, well, actually, so for the predicted age, you can do that. You can't for the mitochondrial interactome, right? Because those animals are sacrificed, right? But because we're taking the tissues, right? No, because this requires basically a whole gastroc muscle. Mouse gastroc isn't that big, right? But so as the third aim of our currently funded R01 is to do this in older adults. So that's the ultimate goal. That's kind of that's where we're going with this eventually. Marry that with the in vivo measures of mitochondrial function, take a biopsy, and look at the interactome. Right. Good. Quadricep. Yeah. So to summarize this last part, mitochondrial protein interactome gives an unbiased view of mitochondrial aging. And I use the term unbiased, in quotation marks, right? Nothing's really unbiased, but it kind of takes a broad view of the mitochondria. And that these appear to be associated with health status and biological age. And so my take-home messages overall are, one, I think we answered the question that targeting the mitochondria to reverse age-related pathology is a sort of an exciting and I think a really an active area of intervention. And we've shown it in older adults and definitely in mice. And one of the mechanisms for this elimipretide activity may be that it's changing this ADP sensitivity, so it's making the mitochondria more able to respond to energy stresses, even at submaximal levels. And this is something that changes in multiple tissues, and so this may be a fundamental part of mitochondrial aging. And then finally, something that the reason I wanted to show these data is I'm super excited about this. What I think is a pretty neat relationship between this mitochondrial interactome and biological aging. So these are acknowledgments. I tried to call out people along the way, but these are all the current people in the lab, and all of them have contributed somewhat in some way to all the studies I talked about. All the collaborators in the Shock Center and kind of around the country, and then so the people that funded the work over the last several years. So I'm happy to take any questions. I did not put in a QR code. Sorry about that. Yeah.
All right. Quick question about the experiments that you're doing with ANT.
Yeah.
So, like the concentration of ATP in the cell is like millimolar. It's really high. So but you were looking at micromolar levels of ADP. Is that physiologic?
That is physiological. So it was a long conversation about it, but really this myth about this high energy phosphate bond in ATP, it's a high energy phosphate bond because it's kept way from equilibrium. So ADP concentrations are kept really low. So resting physiological levels of ADP are 10 to 30 micromolar, and 100 to 200 micromolar are generally max, so about as high as you're going to see in muscle ever. Yeah. But ATP is 5 to 10 millimolar. Yeah.
So, and then kind of relatedly, you're putting out the idea that elimipretide binds presumably to the pore, or is it's something that's keeping the protons from leaking out. So presumably that's a steric. I realize you're not a structural biologist here, but
I purposely didn't go too deeply into the structure because I would quickly get over my head. But
so if it's keeping things from going out, how is it allowing the ADP in?
Yeah. So it's not quite as simple as it's not quite as simple as a cork, right? Which is kind of how I presented it, right? That it's kind of blocking this path. The way that the ANT structure is, it's kind of this hinge. It opens to the matrix, it swings back, it opens to the C state, or the cytosolic state and the M state. Turns out that elimipretide can only bind when it's in the M state. So what we think is really happening is the binding of elimipretide is biasing towards one state or the other, and it's preventing maybe a more time spent in a more open state. So it's not so ANT doesn't work by being a pore and just ADP and AMP kind of passing through it. It binds, brings it in, opens, and then releases it. So we think so we're still working on the real structure. We have to do more we in the people that understand what they're doing, have to do more of the structural modeling. But yeah, so it's not quite as simple as the cork structure, but yeah. Yep.
Yeah, just wanted to pick up on that. Have you or probably your collaborators done any molecular dynamics stuff to model the proton flow? That
Yeah.
shouldn't be too hard from my understanding of
Yeah,
molecular dynamics.
we proposed to do that in a grant that didn't get funded, but that is something that we are hoping to do associated with this current R01. So all the data we're collecting, we kind of want to wait now because we're collecting all this data on changes in the interactome just with normal aging as well as then longer term treatment with the elamipretide. So we kind of want to have all that data in place and then kind of dive into the molecular dynamics. But yes, we propose to do that with Mike Rainier's lab. Yeah, but that would be the logical thing to do.
I have a question. So I have two questions actually. So one, going back into the data that Gavin showed, or that you showed from Gavin, where he is seeing a sort of stair step response with the 1 hour versus the longitudinal time. And so I wondered if you could maybe speak to that, because it seems like you've done it, you guys have probably nailed down what the sort of immediate response is, but I know like in my work at the kidney, we don't see that same immediate response.
Right.
And so what we see is benefit after a long-term, treatment. And so maybe you can speak to what those potential differences are.
Yeah. So this is kind of the model I have in my head, that basically what's happening is there's this immediate interaction between the drug and aspects of the mitochondria, either the membrane or these or specific proteins that can potentially alter function. But then that altered function is going to affect the energy status, the redox status, which then is going to change the downstream signaling. So the data that I pulled out of here, because I just didn't want it to be all over the place, was we've done a lot of work with post-translational modifications and find that longer term treatment, we find changes with age in the phosphoproteome in skeletal muscle with age, and then treatment with elamipretide that restores energy status, restores redox status, also reverses a lot of those changes in the phosphoproteome. And the same thing in the thiol redox proteome. So I think that there's this immediate effect on the energetics that then gets propagated and amplified downstream as same thing for the cardiac function, right? There's less improvement in function in the hearts after a single treatment. I think probably what's happening is you need to do some sort of remodeling of the cardiomyocyte, and that's what you're seeing in the podocytes as well, right? You need to do sort of this longer term treatment to fix more.
And then my second question is sort of related to that, but getting back in sort of to the end of your talk with the sex-specific differences, which I'm super interested in, and I think it's really, really cool. So my question is, have you guys looked at the cross-linking differences just between, because I know that those sort of get analyzed as pairs, and so you're analyzing young to old within a sex. Have you analyzed young to young across sex and old to old across sex?
We have not done that yet, but the way that we've been collecting these and doing the specific, using the different PIR molecules, we're trying to do that so then we can go back and do the old female to old male, young female to young male for that specific reason, because it looks like there's some pretty interesting differences coming up.
Yeah, because the other thing I think we've all seen when we play with elamipretide in the actual animals is there's a sex-specific difference.
Yeah.
But we don't really know what the mechanism underlying that would be. And it seems like you guys would be getting the closest to being able to kind of figure out what that might be. So.
And based on these interactome results, I mean, they're really pretty big differences, or I don't know if they're big yet, but pretty significant and glaring differences between the aging of the mitochondria in the females versus aging of the mitochondria in the males for a given age. So yeah, I think there's going to be a lot to kind of pull apart here. And I only showed these composite measures right now because we're not even at a point where we're actually pulling out, what are the important crosslinks yet, but we're hoping to do that soon. Yeah.
Hi, Dave. Great talk.
Okay, thanks.
I had a question about the remodeling aspect of things. So, I mean, it's fascinating, and it's I used to study anoxia and hypoxia. So the short-term adaptation versus the long-term adaptation is really, really fascinating. And I'm thinking about the hammer I use now and wondering about the transcriptomics and epigenetics, and you guys have probably already done a bunch of RNA-seq on it, I presume, and wondering like what you're finding out, and then what do you see to the other organelles? What the hell are the other organelles doing?
But we have not done very much RNA-seq. We've done a lot of proteomics, but not much RNA-seq. We're in the process of collecting tissues and sending them off for RNA-seq, but yeah, we haven't. Yeah.
Well, far out. That'll be interesting. Do you have any
Yeah.
predictions?
Whether there are going to be differences between the different organelles.
I mean, well, just for the remodeling, like
Yeah,
you expect
well-
metabolic health,
yes.
that sort of a thing.
Yeah.
Do you have any expectations about metabolic changes, or I'm just curious. You're the expert.
Yeah. Where we're going now with this is, what I'm really interested in is how these changes in the mitochondria are affecting the normal adaptive processes associated with skeletal muscle contraction, right? So what I'm expecting, and so the hypothesis that we're trying to get funded, is that with aging, this elevated redox stress, elevated energy stress, just at basal levels, and that's going to blunt a lot of the redox and energy sensitive signaling that's associated with muscle contraction, right? And by restoring mitochondrial function before the muscle contraction, you will restore those adaptive processes, right? So, things like after muscle contraction, things like AKT, AMP kinase, NRF2 is another one we're really interested in the lab. So I think those are going to be the amplitude of comparing a resting to a stimulated older muscle. We think is going to be smaller than a resting to a stimulated younger muscle, right? And then by restoring the mitochondria, you're going to bring that old muscle back up to the amplitude of the response in the young muscle. That's the hypothesis.
Far out. Well, speaking of the mitochondria leaking, do you see pH changes in the muscle cells? Like with age, are they a little bit more acidic? And the cytosol? Okay.
No, not. I think that the pH buffering is such that our ability to measure, so the pH, the way we measure pH is with MR, right? And so that's like taking this volume average pH.
Okay.
So I think if you did see changes associated with proton leak, they would be so local that you'd really have to use a sensitive local dye imaging to do that, and that's just not something we've done. But
Okay. Yeah, I'm just wondering about the mitolyso axis, because the
Yeah.
cells, some cells and senescent cells will get leaky lysosomes, but you can mess up a mitochondria or a lysosome, and both of them cause the cells to become acidic. And that's why I was asking
Yeah.
if there's some homeostasis thing going on there too. But anyway, it's maybe beyond the scope.
Yes. So Mariah's just saying, so Wei Lang, when he was working with Peter, he was looking at the relationship of the mitochondrial pH to external pH, and that does definitely change with age. Yes, with an externally induced pH, that affects the older mitochondria more than the younger mitochondria. So indicating they're leakier. Yeah. Oh yeah. So it's not just our
People have added acidic media to cells, and it causes them to go senescent.
Uh-huh.
But as far as we know, nobody's actually just broken the lysosome on purpose to see if it induces more senescence. So we were just curious about that, because senescent cells have leaky lysosomes, but we don't know if leaky lysosomes cause cells to become senescent. And that's one of the things that is a major senolytic, but there's a big axis of communication.
Nothing
between
we've
them.
done would really speak to that question.
And the reason I'm just harping on this is just because of Keith Nurke's work showing if you break a mitochondria, the cytoplasm of a cell acidifies, which was fascinating to me and not what I would have expected. Right, I thought it would have gotten more basic, but I'm
All right.
Last call. All right. Well,
All
thank
right.
you again,
Yeah,
Dave,
thanks,
for
Bill.
great talk.